Okay, so continuing, this is kind of the part 2, a little bit of continuation of the last video. Before we continue, so we talked about the excitation portion of the muscle. Before we get into the next part of contraction, I first want to jump back to bullet point number 2, talking about the major parts of the muscle fiber and its functions. There's going to be a little bit more anatomy, specifically of the muscle fibers. Okay, so we have our super long muscle fibers, all that kind of stuff. We're gonna zoom in way deep, like way close up of those muscle fibers to look at a little bit more anatomy. We look at this thing that's called a sarcomere. Now, this is always a test question. I promise this is a test question. You're gonna get the question, what is the functional unit of a muscle cell? And that is the sarcomere, okay? The sarcomere right here, this is where all of the stuff happens, okay? Where all the important movement stuff happens, okay? Depending on the professor, you might get a couple of these little things where they're about 2.2 micrometers long, and the sarcomeres, these sarcomeres is what give our muscles that striated appearance, okay? So skeletal muscle is going to have sarcomeres, okay? A couple of the important things to notice. The whole thing is called a sarcomere. We have these different bands, okay? We have an A band and we have an I band, all right? These represent specific things. The A band right here is going to represent the full area. So taking a quick step back, look down here at the bottom. The thick filaments are a protein known as myosin. The thin filaments, represented by these black lines, are known as actin. Okay? Myosin is thick, actin is thin. We'll dive more into specifically what actin and myosin are in a second, kind of what they are and what their jobs are, okay? So we have the whole thing as a sarcomere. The A band includes all of the myosin. Okay, where all of the myosin is, that is the A band. It can include some of the overlapping actin that's there. Okay? So A band, wherever the myosin is. The H zone, this right here, is just where you have myosin. Okay? So when the muscles contract, and we'll look at this all in a second, they all come closer together. The H zone is going to shrink. Okay, the H zone will shrink. Pretend these are our actin filaments, and in the middle of my face is that myosin. It shrinks. The H zone can completely disappear as the actin will come and hit each other as maximal contraction of the sarcomere. We also have this thing called the I band. The I band is where there is only actin. And same thing, as this all contracts, the myosin is going to contract, the actin goes this way, and actin goes this way. So it's kind of like the two myosin get brought closer together. So the myosin pulls this way, this one pulls this way. It's kind of weird to think about it just in like this one little context. You have to look at the whole thing, how the whole entire muscle shortens. The whole fiber, myocyte, shortens. So actually the myosins are coming closer together, and this I band is also, this space is disappearing. So we have the A band, the H zone, the I band, M line. This is where the myosin attach. And then we also have something called the Z disc. The Z disc, these are the ends of the where the actin connect. These are the ends of the sarcomere. A sarcomere goes from Z disc to Z disc, okay? So from one Z disc, the start, to the end is one sarcomere. So here we can see when they contract, you can have two different types of contraction, but you can see, so both ends are free to move, the fibers contract towards the middle, or if there's only one end free to move, the free end of fiber is pulled toward the other. Don't worry too much about this specifics, just understand they contract, okay? And every single one of the sarcomeres contract along the whole muscle for the muscle to contract. This is just a couple different ways that we can picture this. This is a little bit more artistic version of this. Okay, we have our myosin thick filament, the actin thin filament. There's our Z disc to Z disc, the M line, which is between the two myosin. So right here, this whole space right here from the whole thing of myosin would be our A band. Where there's no actin is our H zone. Where there's only actin is our I band. And then this little protein that's right here, some professors like to test on this, some professors don't really care about it. There's an extra protein, it's an elastic-like protein called titin. Titin, its job is to help keep pressure, like elastic pressure, to help everything hold together and help things to go back into place after a muscle contraction, okay? It's also known as connectin, and it restores the responsible for the passive elasticity of the sarcomere. Okay, most teachers don't go too crazy into titin, just usually like what it is, that it's also known as connectin, and that it's responsible for passive elasticity of the sarcomere. That's really as far as they go into it. Okay. Now, we're going to take, so this kind of sums up this little bit of anatomy, sums up point 2, okay? We're going to be done with that here, and we're going to continue into the contraction coupling portion of the excitation contraction coupling of the muscle, okay? There we're still going to be including a little bit of anatomy type stuff as well along with this, but we're going to look at kind of how the muscle is going to contract, and then something that's called the sliding filament theory, okay? So, digging a little bit deeper, we're going to look specifically right here at this actin, okay? So we're zooming in a little box right in here into an actin, and this is what we're looking at. Actin itself is made up of a couple different components. We have G-actin. A G-actin is each of these individual little balls that we can see on here. Each of those individually is a G-actin. When you combine several of those together, then we call it an F-actin. Then we also have a protein called tropomyosin. Tropomyosin wraps around the actin filament and blocks these spaces that are called myosin binding sites. This is where the myosin is going to bind later on when we look at how the contraction component actually occurs. Then we also have another protein called troponin. Within troponin, we also have several components. We have troponin C, which binds calcium, troponin I, which binds to the actin, and then troponin T binds to tropomyosin. So troponin is when it has 3 different parts, and it's kind of grabbing and holding everything together, okay? And remember, in the last video that we talked about, we saw that when we have activation of those DHP receptors and the ryanodine receptors, what got released? Calcium got released. And so then where do we have a calcium receptor? We have calcium receptors in the troponin on the actin filament. So when we release calcium from the sarcoplasmic reticulum, that calcium is going to go, and actually I'm going to see if I can get this whole thing to play, because this little video is all this goes through the step by step. So here we have our actin. Remember, each of these individual guys is G-actin. Each kind of strand is considered F-actin. Those little red spots are our myosin binding sites, and this little thing is troponin. With our F-actin, we have a troponin complex. That should say myosin binding site, not actin binding site, okay? Let me actually fix that. That should say myosin binding site. Okay, F-actin, troponin, the myosin binding site, and this guy right here is the tropomyosin covering up the myosin binding sites. The calcium comes in, it was released from those ryanodine and those DHP receptors. Calcium combines to troponin C within the troponin complex, and that is going to pull and move the tropomyosin off of the myosin binding sites. Now the myosin, you'll see more about the myosin. The main thing to understand is that the myosin, we have these things called myosin heads. Okay, the myosin head is what is going to want to bind to the myosin binding site on the actin in order to generate contraction. So you'll see the myosin is going to grab onto that site, and then we get this something called a power stroke, which occurs. The power stroke is just going to be the actual contraction. Here's a little bit more detail into that myosin head. There's two heavy chains wound together around a tail. So, the two heavy chains wound together form a tail. So these two heavy chains form a tail. And then we have light chains. There's two light chains per globular peptide, which we call the head. The head is what's going to be binding to the myosin binding site. And that is also where we have ATPase activity. Okay, so there's a lot of moving parts. We're gonna at the very end, we're gonna kind of go through a quick summary of all of this. So stick around to the end when we get there. So we have the heavy chains, we have the heads, and when we open up a myosin binding site on the actin, that head is going to have a high affinity, so it really, really wants to bind to that binding site. It's going to bind and do what's called a power stroke. So it's going to bind and pull down. The way that it releases is that we need ATP. So ATP is going to come to bind onto the myosin head, and when it does that, there's ATPase in the myosin head. It breaks down the ATP into ADP, and the myosin head is going to release and then cock and reset. And if the active sites for the myosin, if the myosin active sites on the actin are still active, it'll latch again and pull and repeat over and over again. That actually answers another one of the main bullet points. The energy sources for skeletal muscle fiber contraction. That is our wonderful ATP that we're going to be using. Okay? So, full review of everything is going to go pretty quick. Feel free to pause and come back. So we have an action potential. We have an action potential that runs down the axon of the motor neuron, all the way down. It goes until it reaches the axon terminal. As it reaches the axon terminal, that change in voltage causes voltage-gated calcium channels to open. The voltage-gated calcium channels bind onto vesicles, which contain acetylcholine. That acetylcholine migrates to the end plate. It's going to it's going to exocytose or leave the axon terminal, go across into the neuromuscular junction, binding onto acetylcholine gated cation channels, where sodium is going to come through, potassium is going to leave, and then that signal is going to propagate over and over and over until it reaches more voltage-gated sodium channels across the plasma membrane of the muscle fiber, also known as sarcolemma. It's going to travel along the sarcolemma until it reaches the T tubules, and it's going to go inside of the muscles through the T tubules. On either side of those T tubules, there are terminal cisternae. We call that the triad. And the terminal cisternae have on them, connecting them to the T tubules, the DHP receptors, the dihydropyridine receptors, as well as the ryanodine calcium release channels. Action potential comes through. The change in voltage activates the DHP receptors. That causes the mechanical ryanodine receptors to open, which releases calcium out into the cell. That calcium is going to come and bind onto the troponin C component of the troponin complex. When it binds onto troponin C, a lot like we saw with a lot of our different enzymes and reactions in biochemistry 1, things like hemoglobin, when oxygen binds, you get a conformational change. Same thing happens here. The troponin changes shape and pulls the tropomyosin off of the myosin binding sites located on the actin, aka the thin filament. The myosin heads located on the thick filament are very attracted to the myosin binding sites. So the myosin head is going to grab on and connect to the myosin binding site on the actin, and because it has stored energy, it is going to pull this along. So it pulls the actin along, which is known as a power stroke, and then it's going to have an ATP come along. ATP binds to the myosin head. It's dephosphorylated from ATP to ADP, so thus giving its energy to the myosin head. That's going to break off the myosin head. It releases the myosin head, and the myosin head cocks again, now having taken that energy from the ATP, and it can then bind again and perform a power stroke. So biggest thing here that we need to, that's kind of the whole process very, very quickly. That's what you really want to know for this portion of the material. It's that step by step, exactly how all of that happens. That's all known, if this was to change, that whole process and this power stroke and all this stuff happening is known as the sliding filament theory. Okay? A couple more slides. This is another slide showing kind of that step-by-step just within the actin and myosin, all that good stuff. You can pause to read over that if you would like. Running back to this. So we have now talked about excitation. We talked about the contraction. kind of both separately and how they lead into each other. Next up, we're going to do this in another part video just because this one's getting a little bit long. The actual specific coupling of them, kind of what that looks like. We saw the graph of what an action potential looks like. We're going to look at another graph of the coupling, what the actual muscle twitch and a sustained contraction, all of that kind of looks like, and look a little bit deeper into everything.